Hi, welcome to the BFARM 2018 final year project. This project is mainly looking to identify the genetic drivers of cancer dependency in pancreatic cancer. This is the first video of a series of videos which I will demonstrate to you how you can actually extract the data from the huge genomic project called the DRIVE project, which was published recently in Cells. So we will use this data set to identify the genes that are required for the pancreatic cancer cells survival as well as proliferation. Before we go into the technicality of how to use the Project Drive dataset for our study, I think it is important for us to understand the background of the project. The term drive in the Project Drive is actually a short form for Deep RNAi interrogation of viability effects in cancer. The project was conducted by Novartis, one of the major pharmaceutical companies that produce a lot of the oncological drugs, and the study was actually published in 2017. The main objective of the study was to identify genetic drivers that drive cancer cell survival and proliferation. The way how they identify those genes involve a method called RNA interference screen or RNAi screen. Basically, what they did was transduce a pooled genome-wide lentiviral shRNA into a series of cancer cell lines, incubate them for a certain period of time, and then recover the sequence of the shRNA by next-gene sequencing. By doing so, the shRNA counts for genes that are required for the cancer cell survival would be expected to be lower compared to the beginning of the study, and these genes would then be a good target for development of specific inhibitors to target for a specific type of tumors. What is interesting about the DRIVE project is that it is by far the most comprehensive RNAi library screen reported so far, targeting approximately 8,000 genes in the human genome in about 400 different cancer cell lines. By analyzing the data, it was able to define the common as well as specific cancer dependency genes in different types of cancers. This data, together with other large-scale genomic datasets published recently, will allow us to understand more clearly the biological pathway that drive cancer cell survival and hence lead to the discovery of better drug target for therapeutic purpose. The current study has three specific aims. First, we wish to identify genetic drivers that are required for pancreatic cancer cell survival. And second, we would like to identify the signaling pathway that are associated with pancreatic cancer cell survival. And finally, to evaluate the biological effects of targeting some of those pathways using small chemical inhibitors. To achieve these objectives, we will first identify the new molecular targets from the DRIVE dataset that are required for pancreatic cancer cell survival. We will download the raw data from the Project Drive database, which consists of more than 70 million data points and is publicly available, and pass them through RStudio and Delimit software. The genes that are significantly depleted will be identified using the redundant siRNA activity algorithm, or in short, the RSA algorithm. Next, the signature genes mediating the pancreatic cancer cell survival will be further annotated for gene ontology pathways using Reactome, Davids, and String. In this analysis, candidate targets with annotation result with Bonferroni corrected p-values of less than 0.05 will be selected for future study. At the same time, the gene signature identified from the DRIVE dataset will also be queried against the connectivity map database through pattern matching analysis. The database consists of more than 1 billion data points with genetic profiling of more than 20,000 small molecule compounds and 22,000 genetic constructs for knockdown genes or overexpression genes in multiple cell lines. A connectivity score ranging from -100 to 100 provides a measure of the negative or positivity connectivity of a drug to the phenotype of interest. Positive values indicate that the perturbation gave a similar signature to the query, and negative values indicate that the perturbation gave a signature opposite to that of the query. The mean connectivity score of a perturbation across the 4 cell lines with a magnitude of less than -90 will be considered as hits. Finally, Candidate drugs that are being identified from the connectivity map analysis will be further tested in vitro cell culture model using SW1990, BXPC3, and ASPC1 pancreatic cancer cell lines and MTTSA. Overall, the study is expected to identify new molecular targets for future development of targeted therapy for pancreatic cancer.